Replication is the process of reproducing or copying something. In the case of DNA replication, it is the process of copying a strand of DNA. This entire process is facilitated by a series of enzymes, each with their own unique function, that work together to copy a molecule of DNA. The structure of a DNA molecule is a double helix, with two antiparallel rotating strands of nucleotides with an appearance like a spiral staircase or twisted ladder. The first step of DNA replication is facilitated by the enzyme Topoisomerase. This enzyme attaches to the double helix and breaks a covalent bond between nucleotides in the sugar-phosphate backbone. This allows for the helical structure to rotate, which reduces stress on the bonds of the molecule as it proceeds through replication. Next, an enzyme helicase attaches to a region of the DNA. This is the origin of replication. It then moves downstream on the DNA strand, breaking the hydrogen bonds between the complementary nitrogenous bases. The result of this is a DNA molecule with its two strands separated from each other. Each of these individual strands will serve as a template to construct a complementary DNA strand. Proteins, called single-strand binding protein, attach to the individual strands and help to stabilize them and prevent the hydrogen bonds from reforming. Once the single strands are stabilized, the enzyme primase attaches complementary RNA nucleotides to the DNA strand at the origin of replication. This RNA primer allows DNA polymerase III to attach to the DNA strand because it cannot attach directly to a single strand of DNA. Once DNA polymerase has attached, it can move along the template strand in the 3' to 5' direction, reading the sequence of nucleotides. As it moves along the template strand, it builds a complementary strand of DNA in the 5' to 3' direction by bonding the appropriate nucleotides based on the sequence of the template strand. DNA polymerase must build the new DNA strand in the 5' to 3' direction because, in order to add a new nucleotide, DNA polymerase facilitates forming a covalent bond between the 3 phosphates at the 5' end of the nucleotide and the hydroxyl group at the 3' end of the previously built DNA strand. We already saw helicase unzip the double helix downstream from their origin of replication to the left, but this process would also have occurred moving to the right of the origin of replication, which we can see now. Since the strands are antiparallel, parallel but structurally pointed in opposite directions, the DNA polymerase molecules move in opposite directions because they must build in the 5' to 3' direction. For this reason, moving in either direction from the origin of replication, one strand is the leading strand. This strand replicates continuously in the direction of helicase movement. In our example, the top strand moving to the left from the origin of replication, or the bottom strand moving to the right from the origin of replication. The opposing strand to the leading strand is the lagging strand. This strand is replicated in fragments called Okazaki fragments, as DNA polymerase replicates the DNA molecule in the opposite direction of the work of helicase. So as new DNA is opened, a new RNA primer must be added by primase, and DNA polymerase must then replicate in the 5' to 3' direction until reaching the start of the previously replicated DNA. Once the strand has been replicated, DNA polymerase I facilitates the removal of RNA nucleotides in the primer and replaces them with DNA nucleotides. The final step is completed by ligase, which reforms any gaps in the covalent bonds of the sugar-phosphate backbone like those between the Okazaki fragments. The final product is two identical DNA strands. This process is called semi-conservative replication because half of each new molecule of DNA is conserved or saved and used as the template to build the new complementary strand.